Well, is this working? Good. I am really excited to do this tonight. The only thing that makes me nervous is I've got so much information to share with you. I don't think I'm going to make it, but I'll try. So tonight, we're going to talk about evolution. And God has given us two books. He's given us the Bible, which talks about spiritual things, and the Bible is a closed book. There's nothing added to the Bible since he gave it to us. But he also gave us another book, and that's the book of special revelation, which comes through our witnessing what he's done around us, this creation. And that book is intended to be a foundation for our faith in the Bible as well. And it matters what our understanding of creation is. When I was in college, I had a roommate and I that we were coming back from a youth group meeting and standing in the lobby of the dorm, and a Native American person walked by just as we were doing this, and he was a little bit, had a little bit too much to drink, but anyway, he walked by, and right as we were talking, and so he came up to us and started asking us questions about God. And we said, "Would you like to hear the gospel?" And so I took him upstairs, and we sat down. I was sitting right across from him, and I went through the gospel with him, that we're all sinful and that God has made a provision for that so that we can be saved by Jesus' death on the cross. And you can be saved just by believing in Jesus and, with the desire to repent. And he looked at me and he said, "Well, I have a problem. I can't figure out how God could exist forever." And of course, you might know that that's not a logical contradiction. What would be a logical contradiction would be if there was nothing and all of a sudden all this stuff existed. And so I said to him, "Well, you have the same problem. Where did all this stuff that's around us come from? It had to exist forever if nothing created it." And he looked at me and he gave me this kind of a half-hearted karate chop on my side of my face. And I wasn't too interested in inviting more of it, but I remembered the Bible verse that said, "Turn the other cheek," and so I did. And I just very slightly turned the other cheek, and he saw me. And he got up, and he walked over to the couch and got down on his knees, and we prayed together for him to receive Christ. And so it matters what origins. This first slide that I'm showing here, basically what I've got is these are animals. There's an animal cell there and a nerve ending on the other side. And I'm highlighting things that we'll be talking about tonight. Ribosomes, we'll talk a lot about those. And they're all over the top of the surface of the endoplasmic reticulum that's shown there. And the nucleus right in there has all these little pores that ribonucleic acid comes out of. And this is like the stomach of the cell. It's got acid inside that breaks things down into the little constituent parts that are used other places. And then there's the nucleolus, where ribosomes are assembled with their RNA. And mitochondrion, this is the energy producer in the cell. The mitochondrion takes oxygen that you breathe, and it takes ADP, it turns it into ATP using that oxygen. But I'll show you the process a little bit. And then chromatin, This is the DNA, and it looks like it's just disorganized, filling the nucleus, but it's not. In fact, it's a kind of a four-dimensional organization. The outer portions of it are the ones that are most synthesized, that need to be most synthesized at the moment. And the inner portions less synthesized. And that changes, that organization changes as you develop and as different phases of your life. And then over here you see these little vesicles. They contain the neurotransmitters, and they're actually carried along microtubules to the synapse where they merge. And you can see one right down there in the corner that's actually merging with the wall, and that's how they release their neurotransmitters. And then, of course, this goes off to an axon. Before we start digging into all this, I'd just like to say a prayer. Dear Lord, please inspire our thoughts as I put this revelation of your glory into words. It is you who made the earth by your power, and you established the world by your wisdom. And in your understanding, you stretched out the heavens. Jesus, you created all things, both visible and invisible, from the subatomic to the expanse of the universe. And they came into being through you alone, dear God. Apart from you, nothing came into being that has come into being. Tonight, please make your invisible attributes, your eternal power and divine nature clearly evident to us as you expand our knowledge of your creation, communicating who you are. Lord, we are immersed in the modern synthesis of Darwinism. It's on our televisions, it's in our books, and it's in our workplaces, and so it's so pervasive that it's practically in our food. As with righteous Lot living in Sodom, our souls are tormented day after day by the dogma of evolution. But in your loving kindness, you are opening exclusively to us pages that have until now been sealed in your book of natural revelation, revealing to our generation all that we need to strip neo-Darwinian thought from its foundations and expose its source. Your Holy Spirit explicitly says that in the latter times some will fall away from the faith, paying attention to deceitful spirits and doctrines of demons. Please protect our children, and especially those who are going off to college to raise a family. Protect them from the influences of evolutionary thought. Lay a foundation of observational science tonight for the pyramid of their rational thoughts pointing to you. In Romans 1:20, it tells us that the witness of creation we will see tonight demands accountability to you. Soften our hearts after we hear you speak to us through seeing your majesty and what you've created. What we see tonight renders us without excuse if we do not give you alone the glory. Worthy art you, O Lord our God, to receive glory and honor and power, for you created all things because of your will they existed and were created. Amen. So let's see if this works. Maybe not. It's on. All right. Could you change the slide for me? So don't believe the ad populum ad hominem fallacy. They have names, and because they happen so often. And this is the fallacy. All scientists believe in evolution, therefore so should you. All theories must stand entirely on their own merits. Many scientists believe this fallacy, assuming that other evolutionists have done their work. The supernatural is ground ruled out of science and cannot be included in the theories, therefore they don't come up with any supernatural ideas. All scientists have ulterior motives as well. I know that I have to fight every day against, trying to promote my own ideas about things. And especially if I come up with something really clever, and then I think it's true, but sometimes it doesn't turn out to be true. And I'll promote it, but I have to resist that. Either they earn their living on the theory, or they're completely invested in the theory because they promote it, want to be published, or their reputations are on the line and they bow to peer pressure. Independent thinking is highly discouraged, especially by you if you're not a scientist. 60% of scientists do not believe in God, and the percentage is higher among those working in evolution. God is resisted, and they actively promote that belief and control the narrative. In the preface to What Darwin Got Wrong, and I have these books here so that you can write down, any references that you're interested in. I'm not selling anything. But in the preface of What Darwin Got Wrong, it says, "This is not a book about God, nor about creation. Neither of us are into any of those. Our main contention is that there is something wrong, quite possibly fatally wrong, with the theory of natural selection. Even among those who are not quite sure what it is, the allegiance to Darwinism has become a litmus for deciding who does and who does not hold a properly scientific worldview. We do want ever so much to be secular humanists. In fact, both claim to be outright card-carrying, signed up, dyed in the wool, no-holds-barred atheists. There's only two alternatives. Either God created things or evolution. There's another one. We'll talk about that one later. So let's get an idea of since we're talking about evolution, what it is. It's the idea that mutations of information that's stored in the genome, the fact that it's stored is an important point here, combined with 2 Mendelian genetics, 3 competition or environmental pressures acting solely on the phenotype traits, which is basically the outward traits, the things that you see. And 4 genetic drift produces 5 differential reproduction rates leading to shifts in population genetic makeup or fixation of a single allele. An allele is a variation of a gene. So that came about through the mutation. The Bible says, "For since the creation of the world, his invisible attributes, his eternal power and divine nature have been clearly seen, being understood through what has been made, so that they are without excuse. For even though they knew God, they did not honor him as God or give thanks, but they became futile in their speculations, and their foolish heart was darkened. Professing to be wise, they became fools." And Darwin, at first, he believed. And he thought that nature was evidence for God. But later, he became an atheist. He died mentally ill. If somebody walked by the front of the house, he'd go and hide. I don't know whether that had anything to do with his belief in Darwinism or being tormented by that. But anyway, let's go on to the next slide. Okay, it's working. Very good. This little creature is, it's a shelled amoeba, and I got it from the Sioux Creek. And what it does is it's got little arms, and it pulls them into its, things that it's eating. It surrounds it and pulls it into its body. And right in the center part there, you can see there's an opening there. It's actually dome-shaped. I've got another picture up here too. But it pulls those things in there and digests them. And he's talking right now. He's saying God is creative, all-knowing, loving, kind, all-powerful, master of subatomic as well as the entire universe. The Bible says the heavens are telling the glory of God, and their expanse is declaring the work of his hands. Day to day pours forth speech, and night to night reveals knowledge. There is no speech nor are their words, their voice is not heard. Instead, we and our children see this every day. The heavens are telling the glory of natural selection of the mindless random permutations. Day to day, our media and classrooms pour forth speech, and night to night reveals the neo-Darwinian dogma as unassailable scientific fact. Most scientists believe it. It is fact. It cannot be changed. That actually occurred, that shift where creation was no longer taught as an alternative, happened back in the 1970s and 1980s. I remember when it happened, and all of a sudden people were talking about evolution as fact rather than a theory. So one thing we need to do is make the distinction between historical science, which is evolutionary theories, and the successful observational science that drives technology. They're not the same thing. Observational science, you can test everything. You can't test, seeing a species change into another species. The Bible teaches that we're all product of one race. I forgot to mention that there is a sort of version called social Darwinism that came out of Darwinian thought, and that was Hitler's idea of genocide and eugenics, which was also promoted in America. In fact, Hitler used American eugenics as part of his justification. The Bible teaches that we're all the product of one race, and science bears this out. Genetically speaking, among black people in Africa, there's 85 to 90% genetic diversity. And it's believed that the white people came out of Africa at some point, and we developed a whiteness to us. The difference between white people and black people that we came from is only 10 to 15%. There's more genetic diversity among black people than there are between separating us from black people. So we're all one race, though. And the Bible says in the latter times, some will depart from the faith, giving heed to doctrines of demons. Next slide, please. So why am I talking to you about this? When I was getting my master's degree at the University of Washington, I was working on a project with one of the research doctors there, where we were doing signal processing of MRI images. And he was in particular interested in this one system, a Cygnus system that he had like 2 orthogonal antennas that would increase the signal to noise ratio, because this particular type of imaging is extremely low signal noise ratio. It's like 1,000 times lower than the signal level than it would normally be for a water image that you usually see. And so he sat in the machine for a half hour with his leg sticking in there, and then he gave me the Sigma tape, reel-to-reel tape, and I took it to the VAX computer at Boeing, and I wrote some modem software to receive the data from it and shipped it over to home. And then I wrote all the three-dimensional Fourier transform software to make this image. So it's a different image. You've never seen one of these before. It's an image of metabolism in the doctor's leg. And he looked at it, he said, yep, there's the bone. There's the artery right down the middle. And right there. Anyway, so I work in signal processing and probability and statistics, and for most of my career I've done that. I have some patents in related areas, and I've been working there for 48 years. I also have 7.5 years that I taught Bible at the jail down in Kent. and also Echo Glen with the young kids. I was born the year Watson Crick published their paper, and I put that in the handout so you could see that. And also when the paper, Uri Miller experiment paper was published. So all of this stuff that we're talking about tonight has been revealed in my generation. And your generation is revealing much more than what my generation was. And so, in counter to Darwinian thought, God has given us the most vivid image of what he's created that anybody has ever had. Next slide, please. So here's the first example. In the middle here, you see all the different proteins that are used in producing or maintaining, duplicating, spooling, unwinding the spooling, transcription, repair, all these different processes that are involved in this memory, okay? Evolution doesn't go anywhere with natural selection without all of that stuff. And so over here on the left, you see the DNA. It has to be uncoiled. It's uncoiled by a protein, and then other proteins come along and make the DNA segment that matches up with the corresponding bases. That gets shipped out, but before it gets shipped out, it has to be augmented. There's a gatekeeper at the edge of the cell, and that gatekeeper won't let anything out unless it has this so-called key. And so on the front of the DNA, there's a little protein that gets attached that acts like that key. Also, DNA is designed, or RNA is designed to degrade. And so a number of fake proteins are added onto the back end of it because it's going to get eaten up. And so that protects it. And then outside, there's a ribosome that attaches, it self-assembles around it. And when that happens, then these transfer RNA molecules, and I'll tell you what those are later, but these molecules come in and they decode it. On the one end is attached an amino acid, which all proteins are made out of amino acids. And so that amino acid gets attached, and then the next transfer RNA comes along with its matching segment, and it attaches right next to it. It gets pushed onto it. And then that process goes along until you get this chain. It's called denatured protein because it doesn't have its nature. Its shape, the shape that it folds in, is what determines its function. And so if you don't have that shape, the exact orientation of the active sites on the protein and also the structure that holds those in that position, it doesn't do anything, and it's harmful. Proteins that are not folded properly, one example is a prion, and a prion could actually make it through your intestines into your body. And if you ate cow brain or some kind of brain of an animal that has it, then what happens is that these things reproduce on contact. So it encounters the same kind of protein and it touches it and it becomes a new prion. And what happens is that these accumulate on the nerves and it causes it to be really itchy. And so anyway, misfolded proteins have to be dealt with, and there's a protease that is assigned to go in and destroy anything that's not folded up properly. So there's 1,955 protein types roughly, and this is like an interlocked system with self-regulation. It could not have evolved randomly. And yet, without this system, there is no natural selection. most of evolution has to happen without natural selection. When you get, that big first step, going to the first cell, is a macroevolution step. And that's why I called it that. And so anyway, to avoid having them be misfolded, sometimes they need some help. Sometimes they are able to fold up on the surface of the ribosome, but sometimes they don't. So these are two examples of something that's called a chaperonin. And the one you see on the left is for E. coli. The one that you see at the bottom is for human. What happens is that the misfolded protein goes inside of that. And the lid opens, the protein goes in, the lid closes, and it shakes it up. So it's an annealing process that shakes it up to the lowest energy state. And then the lid opens and out comes the folded protein. And this is the same thing for humans. We have sort of a clamshell thing that opens up like that and it closes. Next slide, please. So we can't cover everything tonight. We're only covering the first three, and there's another part 2. We are first going to talk about the language of protein synthesis. And the first functioning cell could not be explained by natural processes. That's the second item. Evolutionists don't have a good answer for any of these. And they've tried, and I've looked at all of those things to some degree. And so this is my list based on that. The information content of the human genome is overwhelming. Most of you probably have heard of that. The combinatorial capacity of the universe is dwarfed by the requirements of creation. So that's the second part. And natural selection's diminutive influence. And phenotypic decisions are poorly suited for genotypic development. In other words, there's not enough specificity in the outward appearance to go down and make decisions about how a gene should change. And then genetic entropy loses information at 900 times the rate of gain of function. And so on the first topic, next slide, please. We've been talking about amino acids, and here's the chart that shows different kinds of amino acids. They're positive charged, negative charged. You've got peptide bonds between them. They're called amino acids because this little thing up here at the top of each one has this amino acid group. And then it's got these side chains that come down the side attached to the carbon. And that makes them either attracted to water or not particularly attracted to anything. Unfortunately, it's a little bit of a misnomer. They call it hydrophobic because they move away from the water, but it's because they're pushing water away from itself and it's trying to get back together, so water pushes it out. And then the other thing is that the ones that are hydrophilic are trying to get to the water, and so they push it out. So it doesn't care really, but it looks like it's going away from the water. All right, DNA. Everybody's heard of the double helix in DNA. It has a backbone on both sides. It's read out in the order from 5' to 3', 5' being the count 1, 2, 3, 4, 5, 3', 1, 2, 3. And these are the bases that go in between. There's a hydrogen bond that runs down the center, and it's 30 times weaker than the bonds here at the backbone. And so it can be zipped apart really easily, comparatively. And that's good because you want that to survive. So there's 20 of these amino acids, and there's 4 bases. If you assign one base to one amino acid, you could only encode 4 amino acids, right? If you use 2 amino acids, two bases to encode an amino acid, you'd only get 16 possibilities, and that's not enough either. So 3 are used. It's not an accident. 3 are used. It's the minimum possible number that you can assign to uniquely encode all of these, but there's a bunch left overs, right? Because there's 64 different combinations. So those 64 combinations are put to good use, and on the next slide I'll talk about that. But right now, what happens is that this DNA, when it gets unzipped, like we were showing on the previous slide, that gets matched with the messenger RNA. And then messenger RNA then inside the ribosome, like we were seeing on that one slide before, this is a piece of RNA itself. It's like those little crosses, but if you fold the arms behind and you bend it over and then attach an amino acid up here, and then down there are the anticodons that go with the codons over here. That's what you've got here. That's transfer RNA. There's 48 of those. 64 different possibilities, and now we've got up to 48. But there's also, these have the possibility of pairing with more than one different type. So all 64 are used. So next slide. So I put this together from, slides that are pictures that are illustrations that are like this, but I put it in a particular way to show the things that are next to each other. In other words, right at the top there, you've got UUU and UUC. Those are together. You also have, it wraps around. So you've got UUU and over here, UGU. So only one base has changed there. But some, like UUU to GGU, there's 2 changes in that. In communications theory, we talk about the Hamming distance. And the Hamming distance is how many things have to change, how many bits have to change, how many symbols have to change. In this case, how many bases have to change. And so if you look at this, this is not random. And if you think about it, this is the genetic code that makes all proteins. You can't change it. Once you've assigned these things and optimized them, you can't change it, or you kill everything that comes out of it. So it's been optimized with respect to, you see there's groups of hydrophilic versus hydrophobic. It's also been optimized by the distance, that Hamming distance, and the probability of it achieving that level of optimization at random is 10 to the minus 44th. So this is impossible. And let's see, what else do I want to talk about this time? Francis Crick, he thought, this is so complex and it's so pervasive. Almost all creatures use the same code. It's so pervasive that it couldn't have come about by random. So he thought, he's the one that came up with the idea that aliens might have designed it. But that just kind of puts the point off, or the evolution off, to evolve a superior, set of aliens in some other planet. It doesn't make evolution any easier. So next slide. Second point is the first functional cell cannot be explained by natural processes. Next slide. This little thing is a diatom. It has a glass case. It's really made out of glass and silicon dioxide. The glass case has been optimized. It's optimized for 2 things. It eats 2 different ways. It photosynthesizes. and it also digests things. So what it does, it has these little pores on the outside. These are rows of tiny little holes. And I didn't have an electron microscope, so this is what I took. But that looks, it's got a little oil reserve, nucleus and so on. That looks kind of simple. Doesn't look like there's much going on in there. And back in Darwin's day, Thomas Huxley described it as a membrane enclosing a nucleus with various organelles floating in a jelly-like substance. But a single cell is far more complex than that. This chart, and I have a couple of examples of that up here so you can look at it, but this chart shows the chemistry that's going on in the cell all the time, day and night. And there's many irreducibly complex systems that are regulated, and as many as 1,300 enzymes in every cell. And they speed up your metabolism by a factor of 1,000 from what it would be. You would die if you didn't have those enzymes. Those are, by the way, enzymes are proteins as well, and so are hormones. And at any given time, a single cell is simultaneously producing tens of thousands of chemical reactions. We can't duplicate the efficiency of the cell in the lab. And collectively, 100 trillion cells in your body are producing 100,000 different chemical reactions. And then we synthesize 20 to 25,000 genes, different kinds of proteins. They put together a minimally functioning cell, taking pre-existing proteins. They put them all together, and it requires 484 proteins. Evolution can't even do one medium-sized protein, which is about 300 amino acids long. Next slide. So this is an ion, a proton pumped motor. You can see the protons entering from the bottom and exiting at the top. This relies on there being a gradient of protons between this side and this side. And if the gradient starts to disappear, what you'll find is that this thing will stop because there's no propensity for the protons to get from one side to the other. You've got the same gradient. And so when that happens, it stops. And if those things stop in your body, it kills you in about a half hour at the most. That's what happens when you get cyanide poisoning. You're breathing, but you're not taking anything into your cells. And cyanide blocks that. So there's these other proteins. There's 4 other proteins that are proton pumps. And they pump it from this side to the other side. And this one over here also provides electrons to the others for them to pump. And what happens is with cyanide poisoning, this one gets plugged up. And the flow of electrons stops, and then these other 2 can't pump anymore either. You synthesize the same amount of ATP every day as your whole body weight, and they use it. So that's why you don't get twice as big. There's 100 quadrillion of these things in your body. The rotation rate is about the same as a jet engine, a small jet engine, 18,000 RPM. So these things are really moving. And they synthesize 900 ATP molecules per second. This is just 2 views of the same thing. This one shows the structure, and this one shows kind of what the outline looks like. I actually have a picture of it over there that was taken with electron microscope. And this thing has to be assembled in the right order, or it's not going to work. And there's 10 to 15 chaperone proteins that do that. Next slide. Okay, so You don't know it, maybe, but you have microtubules in your body. And they perform some of they do some of the structure of the cell, and they also are the pathways along which this little thing that was walking a minute ago can walk along. It walks from minus to plus. It's called a kinesin motor, and there's 45 different types of them. Here are a number of them, and they carry vesicles containing possibly proteins to different parts of your body where it's needed, and also when this guy walks over here, it's going to have to get to the other side, and so it hitches a ride on vesicles that are going the other direction. And so probably attached to the back end is one of these dining motors that walks the other way. And there's another thing this is a microtubule. There's another type of this, which is the actin filaments and the myosin filaments that are in your muscles. That's what makes your muscles work. These little feet, about as many as a millipede, like up to 200 little feet, walk along there. And it walks a lot faster, 1,200 steps per second. Otherwise, you'd be really lethargic. And the rates, if you scale that up to, walking 3 feet every step, like myosin would be going 3 times the speed of sound. So they really move. Next slide. So this one has to do with the information content of the human genome. So you usually hear from the evolutionary point of view that plants are simpler than worms and fish and so on, and then even more complex are people. Well, next slide, please. Here's a plant, Paris japonica. It has 149 billion base pairs. Our human body is only 3.2. And here's the marbled lungfish, has 133 billion base pairs. If you took those numbers, you can see how many books of 1,000 pages each, like 37,000 pages for that. That's all contained in every cell in that animal's body. Just to preview the next presentation, based on a 14 billion year age, and this is wildly over-exaggerated. The universe itself, the entire universe, could not do exercise more than 5 times 10 to the 140th combinations. And these things are astronomically larger, even in their exponent. So ChatGPT, I sometimes use that. You have to know that it has some limitations to it, and it doesn't do math very well, and it also is biased in its programming. ChatGPT, when I challenged it with this, it said, therefore, the probability of a specific 300 amino acid protein, which is average size, forming in the entire time before heat death of the universe, that's 10 to the 120 years, by chance is effectively 0. And it's widely accepted that natural selection and chemical processes over billions of years of evolution are responsible for the development of complex biomolecular proteins. I couldn't think of a more sarcastic comment that it could say about its own programming. So anyway, only God could have created this system. So let me summarize. Last slide. And I want to give a little bit of time for people to ask questions if there are any. God reveals his power in nature to all of us, believers and unbelievers. It's called general revelation. And I believe God speaks to everyone's heart through his creation. And they either reject it or they accept it, or they just question it for a long time until they figure it out. Historical science endeavors to replace God's message, implying by its name, science, that it has the same authority as empirical science, which it does not. The genetic code for protein synthesis is essentially impossible to have been randomly created. It's one chance in 10 to the 44th. I did that, by the way, by Monte Carlo simulation. I created all these random codes that matched up to the requirements and everything. And I did about 1 million of them. And I compared the Hamming distance of those to the Hamming distance of the genetic code, and it was way off to the side. It was quite an outlier. Okay, a single cell is extremely complex, synthesizing tens of thousands of different protein types. And these are efficiencies that we can't reproduce in the lab. Bacterial flagellum, I didn't talk about that, but it's a rotary motor just like the other rotary motors that generate ATP. And it's, in fact, the flagellum actually can be run by 2 different fuels. And they have, by the way, they're bigger, and they have 11 different gears. And they're positioned on the outside to turn one direction, and they move to the inside to make it turn the other direction. So it can go forwards and backwards with those things. That's something that, it just screams that I was designed by somebody. And it's got all the same parts as a regular motor that we design. Think about the ATP synthesis. It requires an environment where it has ADP in it. And putting the ATP synthesis motors together, ATP synthase, requires ATP to put it together. So how do you get all this ATP in the cell before you have the means of ATP synthesis? There's very little junk DNA available for evolution of new proteins. Most of the 3.2 billion bases are used. In fact, it's about 80% was what the general publication was. And that was determined by ENCODE, which is an international organization that found that they're all being used. And then up to 95% is what they expect that they will find because they haven't finished. So while evolutionary thought has developed more than ever to promote an atheistic worldview, God has countered by revealing to our generation more than any other the depth and majesty of his glory in his engineering excellence. What a privilege we have to be. The Bible really understates God's glory when you think about it. And so, are there questions?
So what do they use to break it down?
Sorry, what?
How is it broken down to all to what you said? I mean, magnification, how to get down to the level of things small.
How can you see it? How can you see these processes?
How can you see the processes?
I mean, having all the cells defined as it is, how do they know? I mean,
how do they know it? These things that I showed you, that's thousands of researchers spending their whole life.
But how do they determine that?
Oh, how do they determine it? Well, obviously the electron microscope. You can see the layout of things with the electron microscope. They also look at concentrations of different things. So they'll have it synthesized and they'll look at the change in concentration. And they can, of course, they can sequence the DNA. And then just lately, the University of Washington, one guy at the University of Washington and a couple of guys at Google won the Nobel Prize for their work in synthesizing protein. and they can actually synthesize a fairly large protein to meet designer requirements. But they have challenge with some of the ones the size in the human body. But that work was just recent that they did that. And by going through that kind of process, you start to learn about the process itself.
So they can physically see that?
They can physically see the protein, and they can see it on their computer screen when they're designing it, what shape it's expected to be in. Google did the AI research that took a lot of different proteins to see how they folded and trained it on that. And then this guy at the University of Washington then developed a system that allows you to use that AI result to design a protein, see what it should come out to. So yeah, they can see it sort of in different ways. Any other question? Yes?
Where do you find those animals you showed us at Sea Street?
Oh, the animals that I showed, that was the diatom, yeah, and they're all in the mud. And you just go in there and you take a jar and you scoop them up, and you put them on a slide, and you can see them. And I've seen water bearers in there. There's little creatures with 4 legs that walk around. They're clear, and those things are amazing. What those things, they can live in outer space in a vacuum, and they also withstand really high temperatures and stuff. So there's pretty amazing things you can find just in Sioux Creek. But you can find it in mud puddles too. Sorry, what?
How much is your microscope? The microscope you use?
Yeah, I have a metallurgical microscope. It's about like that. And that allows me to look at the surface and also look at the transmission of light through it. It cost me about $1,000, I think. Dar, you had another question?
She answered my question.
Oh,
I said, what was the original question?
go ahead.
No, we were just figuring out what Daniel asked.
Oh, okay. All right, any other questions? Yeah.
So you talked about like the processes and how the gears work, and there's transfer of protons.
Yeah.
I know it's not necessarily within the scope of this, but on the metabolic waste side of things, do you have any kind of quick thoughts on what does that represent, or what does that say? Like how the waste processor.
Oh, how the waste process is in the cell. Well, there's a lot of waste in the cell because proteins are being misfolded, and they're just junk floating around sometimes that you can't use. And what happens is that those vesicles that I was showing, where the thing was walking along the microtubule, those things sometimes will carry those wastes to the lysosome. And then the wall of the vesicle merges with the wall of the lysosome, and that spills its content on the inside. And it has acid. It's got like proton pumps to make it acidic inside. And that acid then breaks it down into its constituent amino acids and bases, and then those can be reused, and they're expelled from the cell just the same way. And then also there's parts that you can't do anything with, and they're put into a vesicle and carried to the outside of the cell, and where it merges with the wall, and out it goes. Okay.
One more question.
Oh, yeah.
So it's more of a philosophical question, I guess. But they say people are crazy to believe in creation, but scientists are really smart people, theoretically, right? And
Sure.
so looking at all the proof, and it seems a lot more logical to believe that something that we were created rather than it's all random. So what is there to gain for scientists to argue against creation when it seems more logical to that we were created as
Right.
opposed to it being random?
Right. People generally believe what they want to believe, And scientists working in that area of evolution, they want to believe in evolution. And so they'll pick facts that, that support what they believe. But the thing is, they pick a lot of facts that don't matter. These facts do. And they put their emphasis on those. There's a lot of arguments for evolution out there, seeing changes and things like that, they have several different animals that they point to and say, this is a transitional animal. But that doesn't matter because this can't be explained.
Yeah, and I just don't understand what there is to gain to argue against it, against creation, I guess.
Yeah.
I guess it's a hard question to answer.
Well, each one of those things that I listed has a counter argument by evolutionists. And there's like 7 different counter arguments, or basically ideas of how chemical evolution could have happened. And the guy that wrote the book, one of the original books on chemical evolution that was used in the classrooms, he later on said it didn't happen. So once you get to the point where you're willing to accept God, then you can start to see that God did it. I do have a part 2, not tonight, but there is a part 2 where I'm going to talk about the weaknesses in the theory of evolution. Tonight was all about God's creation and how amazing he is in what he did. Oh, there's a question back here. Yes. And the question was, does everything have a DNA in it? And the answer is yes. All animals have DNA, and that DNA is what maps out the development of that animal and what maintains it. Gets all the instructions to maintain it is in that. Yeah, even plants. Yeah, go ahead. Why is it coiled like that? Okay, so each of those little atoms in the molecule, they have their particular bond angles that they like to have because of the orbitals that are in there. But that's the quantum mechanics part of it. But they have these preferred, like oxygen and 2 hydrogens. It kind of makes a V shape like that. And with the oxygen at the top, that shape that's water, right? Oxygen, hydrogen. And you won't find water in there, but there's all of those bond angles determine that this thing will twist around itself. That's how it does it. Okay. Up here on the stage, these are a number of books, just some of the books that I have in my library that I use that talk about that. You can write down references. This chart right here is the chart that shows the cellular and molecular processes that are going on in cells. You got that? Okay, all right. And this one over here is the chemistry of the cell that's going on in there. So if you go to the Roche site, you could see this, except right now they're updating it. So you can't see it, but soon.
So in short, you're saying that mathematically the evolution can't happen?
Yeah, it definitely. That's it.
All right. Let's go. Hold on, guys. Tom, thank you so much. I'm so glad there's no test after this. But more importantly, thank you that you showed us just the complexity of a cell and how God has made everything. Really is amazing.